Okay, so welcome back everybody to REYES. This is the last week of REYES, so thank you so much for sticking around for 8 long weeks. If you, for some reason, are not able to watch this live streams, just remember all the videos that we recorded and all the material that we've presented to you throughout these 8 weeks are going to be made freely available for you indefinitely. So you're going to be able to enjoy these videos, not just right now, but also in the future. And so with that, I'm very excited to introduce our next speaker, Dr. Terry Grant. So Terry is a colleague and a friend. She's one of our success stories here at Old Dominion University. She got her PhD in 2008 in computational applied math at the university, as I mentioned. She's going to tell us about her research, which is quite interdisciplinary. Although it's in the field of applied mathematics, is pertaining to chemistry and biochemistry. So she continues to do part-time research in collaboration with these departments, and she's a full-time lecturer in the mathematics department here at Old Dominion. And so with that, thank you so much, Terry, for taking time out of your schedule to tell us a little bit about your research. So I'll leave it up to you. And so thank you again.
Thank you so much. Hello, everyone. I just wanted to share a little bit of my research with you and how you can take 2 or more STEM branches and combine them in order to look at research for real-world situations. So in this case, we want to combine chemistry, mathematics, and coding in terms of estimating the 3-dimensional structure of a protein. And so let's go ahead and get started. So when we think about a protein, we normally think about something that's in our diet, like a good source of protein would be meat or fish or broccoli. However, proteins by its definition is a large complex molecule which our bodies and the cells in our bodies need to function properly. So proteins come in many different sizes, and the proteins are found throughout our body. So this is the chemistry aspect of this particular type of problem that I worked on during my research. Proteins are often referred to as the building blocks in the body because they're a central role in growth and development. And so just a few types of proteins that I thought about as I was developing this presentation was some of those keywords that we hear when we turn on the news: antibodies. We are probably familiar with the pandemic that's going on now. So when we hear about antibody testing, we say, well, what does that mean? Or hemoglobin or digestive enzymes. These are all different proteins and they have specific functions. So the antibody, key word here is defend. Antibody is a protein that protects the body from foreign substances such as bacteria, viruses, and other microorganisms that cause disease. And so here, application-wise, why would we want to know about proteins or modeling proteins or estimating the 3-dimensional structure? Is because it gives us insight into how to attack diseases or cure diseases. Another, hemoglobin, which is a protein that transports. And here what it does is it carries oxygen from the lungs to the body's tissues, and it returns carbon dioxide from the tissues back to the lungs. And here in this picture, what we notice is the normal shape of a hemoglobin molecule. And what happens if the shape is not correct? We end up with a disease known as sickle cell anemia. And so the shape of a protein is very important in terms of being able to cure diseases. Another example of a protein, digestive enzymes, and their work or their job is to decompose. So for this type of protein, it breaks down nutrient and food into small pieces that can be readily absorbed in our body. And so when we ingest food, the food goes into our body and then it's broken down and then it's used for what it needs to be used inside of our body. Now, with that being stated, the shape of the protein is the 3-dimensional structure. By folding into its unique 3-dimensional structure, a protein, we can determine the protein's biological function based on predicting it based on structural similarities of closely related proteins. In addition, we can determine which molecules or drugs can efficiently bind and how they can bind into a protein. And lastly, we can gain more important information about diseases that cause from protein misfolding. And so because of the fact that it is important to be able to identify the shape, we want to understand the level of which the protein folds. When a protein folds, it folds in levels. And so if we're familiar with any level of chemistry, then we're familiar with the different levels of protein folding. Primary structure is called the primary structure because it talks about the protein in terms of its sequence of amino acids, which are held together by peptide bonds. And there are about 20 amino acids, and the amino acids have in common certain pars, but they differ in certain pars. They have the amino group, the carboxyl group, as well as the hydrogen and the carbon. And the R group is what distinguishes one amino acid from another. But the chain of these amino acids is what creates a protein. And so that's why it's called the polypeptide chain. Now, when there's some types of interactions within the sequence of amino acids, hydrogen bonding, disulfide bonding, then we end up with secondary structure. And so with the secondary structure, that arises from hydrogen bonding and between the local groups in the amino acids. And then from there, based on the types of side chains that the amino acids have, if the amino acids have the property that they're hydrophobic or hydrophilic, then that will determine the tertiary structure. So the basic understanding of how a protein folds is that from the sequence of amino acids, the secondary structures form first. That would be the here alpha helices or the beta sheets. There are other types of secondary structures, but these are the major ones that are really recognized in many proteins. After the secondary structures form, then the secondary structural elements pack together into a tertiary arrangement. And that tertiary arrangement or tertiary structure is what we're referring to as the three-dimensional structure, which is determined by all the other interactions between the amino acids within the sequence. Lastly, we have the quaternary structure, which is two or more polypeptide chains bonded together. And so in this particular presentation, we're going to focus in on predicting the three-dimensional structure of a protein, or estimating rather. And so when we think about this type of problem, this problem is not just solved by one group of scientists. It's solved by scientists whose background is in chemistry or biology. It's also solved by scientists whose background may be found in physics, mathematics, as well as those in the computer science area. And so when we think about this type of interdisciplinary type problem, it encompasses all the different branches of sciences coming together and working together to find a solution to estimating the protein's three-dimensional structures. There are other aspects of modeling protein. One can model the secondary structures as well as look at the behavior in different amino acids within the protein. But here in my research, I spent a lot of time using computers and algorithms to be able to estimate the three-dimensional structure. And so ideally what happens is, in order to be able to determine or predict the three-dimensional structure of a protein, normally experimental techniques are used. And those experimental techniques, the major ones are X-ray crystallography, as well as the nuclear magnetic resonance spectroscopy. And so the first experimental method is where X-rays are aimed at the solid crystal of the protein. The crystal is then scatters the rays onto a detector that captures the three-dimensional aspects of the crystal structure. And here for this particular method, it's limited in the amount of time required to crystallize the protein. The difficulty in determining all of the information or the three-dimensional structure using this technique is because for large proteins, it takes an extreme amount of time to be able to determine that information from this technique. So the second technique here, NMR Spectroscopy, this particular technique is based on solubility behavior of the protein. In this case, if the protein is soluble, the protein is submerged into aqueous solution and then analyze the relationships between the magnetic field and the atoms that are commonly found in the protein, which would include the carbon and nitrogen. However, there are some proteins that are not insoluble. So in recent years, there's been a lot of work in developing computational algorithms that will help in estimating the three-dimensional structure based on the information that we can gather from experimental techniques. And so all of the information that's gathered from either experimental technique or those that I haven't mentioned here are deposited into a data bank, and that data bank is known as the Protein Data Bank, PDB data bank, or database rather. And in that database, it contains information about the structure, the coordinates of the atoms within the protein structure, the sequence of amino acids for the protein structures, as well as some of the other types of chemical information about the structure. With that given information, here at ODU, I want to mention that for those experimental techniques, we do have a major instrumentation cluster that, again, a lot of chemistry students do predict the protein structure using these different types of instruments that are located here at ODU. But for myself, the way that I work through solving this type of problem can be given in this flow chart here. And so the idea is we start off with free input data. That data comes from the sequence, comes from structural data, and the data that we're looking for can come from any of these types of methods here: secondary structure prediction, homology modeling, experimental methods. And so all of that information is taken, and we use that information, distances, angles, volume, surface, and we develop a formulation. And once we have this mathematical formulation, it's not something that we can do by hand. We need a nice, efficient algorithm that will perform and allow us to be able to use this information here in order to be able to estimate the three-dimensional structure of a protein. And given the three-dimensional structure of a protein, why is it important? Quick and simple. If we know the structure, there are ways to determine the biological function. And so most of the time when we talk about modeling aspects can come in any of these regions, where for me would be in this region here, where we're given some input data that's gathered from different experimental techniques. We improve the formulation, maybe improve the algorithm, and use the improved formulation and the algorithm to model proteins, three-dimensional protein structures. And so let's talk about the three elements of estimating protein structures, the three-dimensional structure. First, there's free structural data. What makes the data free? Where do we get this data from that allows us to set up this formulation? What is the mathematical formulation? There is more than one. And what are the types of algorithms and programs that one would use in order to be able to find this three-dimensional structure? And so let's start off with our structural data. And so when we talk about structural data, we want to be specific. Yes, we know that it's distances, we know that it's angles, but we want to make sure that the problem is realistic to what we're modeling. And so here's just the basic geometry of the protein's atomic representation. And here we notice that we have the amino acids, which is defined by here the NC alpha C. So we have this side chain. Now here the side chain is represented as R, but the side chain takes on many different atoms for any given amino acid. So what we want to demonstrate here is just the types of information that we look to use in our algorithm or our formulation. And so here we start off with just the main chain atoms, and from here we're looking for the bond lengths. We may be looking for local non-bonded distances, non-local non-bonding distances, which we can see here in this picture, as well as angle information, the bond angles, which are generally rigid, torsional, or what we refer to as dihedral angles, which are important to forming the secondary structures, the major secondary structures of the protein. And so here again, the first way that we can generate all of this information is based on the experimental techniques. Go to the protein data bank, and there we'll find information about the proteins that have already been the structure for which the structure has already been determined, and then we can use that information. Another way to gather structural data is from homology modeling or comparative modeling. And here the basic idea is we're given a sequence of alignment of a set of reference proteins. The structural data is extracted from the conserved regions of the set and is used to be able to estimate the three-dimensional structure of the target protein. So here is just a sample of a sequence alignment. And remember here that these are proteins that are known and they're part of our PDB data bank, which we know the sequence, we know information, we know the positions of the atoms within this protein. And what we're looking for is information about the conserved regions. And here, the uppercase letters represent the amino acids. And so we can kind of see in some of these regions where we have commonality in the different types of amino acids. But if we look at the reference protein, for this particular example, we already know the structure of 1-CTF. But if we look at the reference proteins, the proteins that are closely related to this protein, we can see how similar the structures are. And so in terms of generating structural data, if we did not know the structure of 1-CTF, then we could use its closely related relatives and find some of the structural information using statistical analysis and use that information to determine the three-dimensional structure. Another way of getting structural data is to repurpose modeling tools for other aspects of protein modeling. And what does that mean? There are a lot of other modeling programs that are available that are not necessarily being used for estimating the three-dimensional structure. One of those is molecular surfaces. So here, the basic idea of forming the molecular surface is that we have a molecular surface around our protein, and what that does is it allows us to detect possible drug binding sites on the surface, identify the possible proteins for pharmaceutical use. But in terms of estimating the three-dimensional structure of a protein, we want to be able to model the hydrophobic, hydrophilic interactions of proteins. Now here we have to be careful about what types of proteins we're talking about here, because there are many types and some are more hydrophobic and hydrophilic. So in this case, we look at what we call globular type proteins, which have this type of interaction as part of its folding process. And so remember, the basic understanding or assumption of protein folding is we have a sequence of amino acids. From the sequence of amino acids, we assume that the secondary structures form first. That would be the alpha helices, the beta sheets, the loop regions. And then from there, the secondary structure elements will pack together into a nice three-dimensional structure. And so to model that, what we use is the surface to give our secondary structure elements correct proximity to each other. And so with that being stated, there are a lot of other different ways to gather data. Those are just examples of how one would use structural data from different experimental techniques or different modeling techniques and use into a computer algorithm. So now let's talk about the mathematical formulation, because here it's like, okay, I'm a math person. I'm not chemistry, I'm not biology, I'm not physics, and computer science, I'm not that either, even though I can code. Again, here, when I look at solving this problem, my job as a mathematician is to be able to improve the mathematical formulation. What works for a mathematician may not be what works for computer scientists or what works for a physicist. You say, well, what does that mean? What that really means is what works in principle may not work in practice. So let's take a look at the mathematical formulation, and I can explain a little bit more. So mathematical formulation, the primary way to assume proteins fold is that, or when they have their functional, biological functionality, is when the protein is at its lowest energy state. And if we know anything about physics, that's normally a measure of the potential energy. So we have the potential energy formulation. We also have the knowledge-based formulation, which is kind of closely related to the comparative modeling, where we have all of these proteins that are related to a target protein, and we're looking for structural features that are in the conserved regions. What's the probability of a specific type of structural feature occurring in a given protein? And those are called knowledge-based functions, probability distribution, or density functions. And then we have also the formulation of nonlinear least squares. And this is where we measure the error between the input information, the structural data that we're given from the different experimental techniques, and that we would get from the three-dimensional model that's created from our formulation. And so we have all these different formulations here. And so it's not just one formulation working. It may be one, maybe just a program that's developed based on just potential energy, maybe a program that's just developed based on knowledge-based information, and then one that's based on nonlinear least squares. But most of the time, all of these formulations are combined, because if we have all of this free information, then we want to use all the information, whether we're using this formulation here, the first formulation, the second, or the third formulation. And so when we think about the words that are highlighted here, lowest energy state, we want the lowest state, and this is where the protein is biologically active. Here, when we think about error between the input data and the three-dimensional model, we want the error to be small, because if the error is small, then we know that our model hopefully is working correctly. And so those keywords lend itself to one specific type of formulation, but whether we're looking at the potential energy, knowledge base, nonlinear least squares, the fundamental math problem is the same. Find the coordinates of all the atoms in the protein, which is referred to as a configuration, that best satisfies a set of structural data. Now, whether that structural data is just distances and angles, surface information, again, the problem is the same, to find those coordinates that best satisfy the data. So with that being said, one of the different types of math problems that we can think about as related to modeling the three-dimensional structure is here what we call an optimization problem. And so when we have an optimization problem, we have several different components that are important to the setup of that problem. The first is the objective function, a function that measures how well a given configuration satisfies the structural data. And what we want to find is the solution by minimizing the value of the function. And so here, the objective function would be a function that's either potential energy, least squares, measures least squares error, or either statistical potential. The decision variable is the variable for which the objective function reaches its optimum value. And so in this case, we're looking for the configuration or the coordinates for all atoms in the protein. Configuration space. Here, because we're minimizing our function, there's more than one solution that could work for this type of problem. But what we want is the optimum solution. Here, the word optimization. We want the best solution given the set of structural data. So we have the configuration space, which is space of all possible solutions for which the function can be minimized. And here, we have constraints. When we think about minimizing a function, and let's just look at distance information. If we minimize a function using only distance information, we can kind of figure out what's going to happen to minimize the function value to be close to zero. It's going to end up being some structure that's not biologically plausible. So we need our constraints. These are limitations on the configuration space that are determined by the physical nature of the problem that we're solving. So here we're looking to estimate protein structures. And so some of the physical characteristics that we want to be aware of is minimum separation distance. And that occurs when we have atoms that repulse each other, van der Waals, as well as chirality. When we look at chirality of a given protein structure, certain elements within the protein can take on one of two different positions, what we call the left-handed position or the right-handed position. And here, we want to be able to make sure that our protein structure satisfies these type of constraints. And so one way to do that is first, let's look at the terms in the objective function. What are the terms that we would include in this function? A term that models all the different aspects of chemical behavior that one would find in protein folding. So bond stretching, or least squares error function, is given by this first equation here. And here our S sub ij is the stretching constant, or the reciprocal of variance. And here we have our input information and the model, the calculation from the model. And so ideally what happens here is that this constant in front of this difference squared is represented by either, again, the stretching constant or the variance. How reliable is the data? If the data is high variance data, then we might not want to use it. Or either we want to have a measure that says that it's high variance data. If it's low variance data, then definitely that's data that we want to use. So in a least squares type of formulation, that's what this would represent, is how accurate is our structural data. Other types of functions or terms would include the bond bending, which is our bond angle. And here again, just these are the measurements here, and this should read as the known angle and the calculated angle. But normally when we look at angles, we can try to use distances in the place of angles to be able to model the same aspect. It doesn't always work, but again, that's one way to model angle information, especially if we have an objective function that deals with angle information. Electrostatic potential, as well as van der Waals potential. And so here, these two are to model the repulsive and dispersive behavior of atoms within the molecule. To ensure the overall volume is maintained, the constraint forces a separation between the side chain atoms and one residue with the side chain atoms and another residue. In that terminology, residue we refer to as amino acids. So it forces a separation between the atoms in one side chain of an amino acid to those in the side chain of another amino acid. And here, torsional angles. Remember, torsional angles are the angles that are important in forming the secondary or the substructures of the protein. And here, what we normally would do is determine the detailed features by using distance input. But when we use distance input, that brings into the picture the mirror image configuration, which we might have the incorrect orientation of the substructure that we're interested in modeling. And that leads into another type of constraint, which is called chirality. And so when we look at the chirality constraint, it requires at least or 4 specific atom types to enforce the left-handed, right-handed orientation of substructures within the molecule. So a lot of the substructures tend to fold in a right-handed type of orientation, but when we're modeling using distance information, it may form these substructures in a left-handed type of configuration, as opposed to the correct structure that it should form. So here, the other part that we look at is enforcing physical conditions, physical constraints. And one of the ways to do that is to introduce another term into this potential energy function. And that additional term is included in the objective function, and it assigns a very large function to the function value to the objective function for any given atom placed where the atom is placed in the violation region. And so some of those constraints, again, minimum separation distance, van der Waals constraint, chirality constraints, torsional angle constraints, and our surface constraint. And there are other constraints that are not included here, but again, we want to be able to model all the different types of behavior that we see that occurs within the protein folding process. And so there are 2 different ways that I see this formulation of estimating the 3-dimensional structure. As we call it, an unconstrained optimization formulation. And what that says is find a set of coordinates of all the atoms in the protein that minimize based on minimizing the following function, where the terms in the function model certain aspects of our protein, whether it's the structural aspect or whether it's the chemical behavior that occurs between atoms within the protein. The other formulation is constraint optimization formulation. And you say, well, I thought these were the same. We have constraints, we have an optimization formulation, isn't the same. But from the perspective of one who's in the math field, it makes a difference because determined upon how this problem is set up, we can identify the best algorithm to use to solve the problem. And so here we have this n-dimensional, n-variable type function with multi-terms. Picking an algorithm that's going to work to minimize this function is going to be difficult. But for this particular, the second one here, we have our finding the same decision variable. But if we just choose a simple interatomic distance type function, and then we try to figure out ways to introduce the other constraints, not into the objective function, but into the problem or the algorithm, then it will make it maybe easier to find the correct or the optimum positions of all the coordinates of the atoms in our protein. And so here we have the two statements, whether we're looking at the first or the second formulation, it's just a matter of where our constraint information is placed. Which one is better? There are a lot of different models and software packages that use this first formulation here and just work on developing the algorithm. If I use this function and I find the best algorithm possible, I should be able to come up with the correct protein structure or close or near accurate protein structure. This one here says, well, let's work on developing the mathematical formulation and then use the algorithms that are available, whether it's the best algorithm or not. If we have a simple objective function and we think about ways to introduce the other physical constraints that makes the problem more realistic to what we're modeling, then it will make it easier for us to find the minimum based on the function. And then from the function, check and see if our solution violates any of these types of physical constraints that we see. So let's talk about the algorithm. The third part, the first part was just the structural data. The second part is the mathematical formulation. The third part is, now that we have this mathematical formulation, what type of algorithm do we want to use to solve this problem? And so here I wanted to take a step back and think about if I had seen Calculus 1 or Calc 2, and in that course, we looked at being able to solve optimization problems. What are we really looking for? We're looking for the minimum, in this case, the global minimum. But most of the time when we're looking to find the global minimum, if we look at the objective function compared to its configuration space, there are many different types of local minimums that can occur. We can have the flat local minimum. In this case, a local minimum. We have the global minimum. And here we have what is called a shoulder, but it could be considered a max or a minimum. So when you're thinking about the algorithm is supposed to be able to not only find the local minimum, but it's supposed to also be able to find the global minimum from among the local minimum. And here's the issue. Based on the formulation you're starting with, it may be a problem. Why? Because if we're starting with a formulation that's a nonlinear, multi-term objective function, where we have a function that has 3 n variables for the number n represented by the number of atoms, then trying to use algorithms to solve this problem is doable, and it does work. It's been proven that it does work, but there may be a better way to approach this. But with that being stated, we want to look at the different options that we have available for finding local minimum as well as finding global minimum. And so here, just a standard form for a local optimization algorithm, these are also called local search algorithms, is to start with some initial guess, initial current starting configuration. And then from that starting configuration, we use some type of update procedure to find the next ideal position for the decision variables or for the positions for our X and Y coordinates or coordinates of our atoms. And then we compare the function values. If the function value of the next is greater than the function value of the current, we return the current. And what does that mean? We want to return the lowest function value because we want to minimize, whether it's potential energy or minimize error, we want the lowest function value. If this is not the case, we set the current equal to next, and we continue this process iteratively. We consider this process over and over again, and at some point there's a stopping criteria, and then once we have that stopping criteria, we can choose the better configuration, or randomly choose what the better configuration would be. So to explain that without looking at words, the best way for me to explain it is through how do we update our configuration. We start off with a given configuration, and then we update the configuration by looking at not only this alpha, we look at the change in x. And here, the change in x is the step direction for which the function decreases. Alpha is the step length. Once I find the step direction, how far along that direction do I move to get to the next optimum solution? And so the process of finding the alpha and the change in x is what's referred to as the local optimization method. How do I find or choose these values so that when I choose these values, I can find an optimum solution for my algorithm? And so here we have to be aware of the number of equations we have. If we have n atoms and we have here 3 times n coordinates, when we look at solving these types of local optimization problems, we want to be aware of the different techniques we have available and which would be the better one. So local optimization algorithms that are being used, gradient-based updates, which uses the first derivative information. And so the best way for me to explain this is if you think about a function, a single variable function, and you think about how do you find the local minimum of a single variable function, what you would do is you would compute the first derivative. Once you have that first derivative, you set that derivative equal to 0. That will give you what you call critical numbers. And then you would look at the curvature or the behavior around those critical numbers to determine whether your critical number is truly a local minimum, local maximum, and so forth. And so that's what these algorithms are designed to do, to be able to determine the curvature of the function around a given choice or update. And so the gradient-based method is normally used because it's easy to implement and because it doesn't require a large memory space here to actually compute the first derivative information. But it's not the most accurate depending upon the type of problem that you're solving. If you're solving this type of problem, protein estimation, then Newton's method update is normally the one that's used, or a quasi-Newton type of update. And what these update procedures do is they identify the step direction needed to update the position of the configurations. After we have the step direction, we can use what's called a line search to be able to identify the step length, how far to move in the direction based on other information or the setup of our objective function. And so here, when we identify minimum, going back to this picture again, what the local optimization algorithms are going to do is they're not going to find the global minimum. What they're going to do, they're downhill type of searches, so they're going to find the closest local minimum. And in this picture, it would be this minimum here. And so when we get to this minimum point here, if we start with the local optimization type algorithm, it's going to give this as a solution. Now here, in looking at local minimum, I guess a good question is, where does the local minimum occur in a protein folding estimation type problem? Local minimum comes from the type of function we're dealing with, the type of optimizations that we're using, as well as satisfaction of the physical constraints. And so we don't know what type of local minimum this would be, but what we do know is that we want the global minimum, the optimum position of these values, which would be our global minimum. And so in order to find this, one way to do that is to say, okay, here are all the local minimum. From among the local minimum, I should be able to find the global minimum. And there are methods for that as well. Global optimization methods, those are deterministic methods, as well as stochastic methods and our last type of methods here. And all of these methods are designed to be able to find the global minimum. Now, it depends on the number of variables, how reliable these will be, but the one that's most used or recognized is the simulated annealing process, not the one that I've used, but it definitely does work, or the stochastic methods because it takes a random step and it searches more of the configuration space. And so here I won't go into any great detail about the different benefits of each of these methods or the disadvantages of each of these methods, but for certain types of problems, these methods here work better depending upon what the problem is you're trying to solve. And so here, back in this picture, we look at what a global optimization type method would do. It would search more of the configuration space. And so where the local minimum would end up here, a global optimization algorithm would say, well, let's try to check some more of the configuration space and see if maybe we can find another local minimum or the global minimum. And so here, if it's a random type, perturbation type technique, such as simulating annealing or those stochastic processes, then here it will just search a lot of the space. One would think that once we're here, it would just converge to the global minimum, but it doesn't. Depending upon how it's set up, it may converge to another local minimum, and at some point find this local minimum, and then eventually it may move and find the global minimum. And so this is all the different types of parts of an algorithm that should be considered when trying to solve this type of problem, is which one will really work to find the global minimum. And so the methods that work here have been used and are successful, but there are some challenges. Some of the challenges: estimating the three-dimensional structure of a complex protein from sparse and noisy input data leads to difficult nonlinear optimization problems. Sometimes it's very difficult to include specific interactions in the objective function that can determine the detailed features of the substructures, such as the secondary structures. The optimization method that's being used, or the algorithm, may yield poor quality structures due to local minimum or misplacement of placement of atoms in positions that violate physical constraints. And so what are the metrics? To finish this off, the metrics would be, how would we really determine whether our algorithm really works? There are a few metrics, but one of the main ones, of course, is the low function error, as well as the root mean square deviation is used to measure the accuracy of the structure that's obtained through this type of modeling. So this first one says the function is minimized. That's what the low function error means. The second measurement gives us an idea of how similar our structure is to the original structure. And so here I'm just using the substructures within the protein to demonstrate here, but we can see here in this case where we have a low function error, high root square mean deviation, or RMSD, we can see here how similar the structure is, but we can see that it's not identical. And so the two together, not only is it enough to say we have a low function error, because with low function error, there are many ways we can minimize a function and the structure not be identical or correct. So here we have low function error as well as low RMSD. And ideally what we want to happen is we want to have a nearly identical type of conformation. And here again, this is the secondary structure, so we're not looking at the complete three-dimensional structure, but just as a visual of how these metrics are applied. In this last one, we have high function error, high root mean square deviation. And here in this case, the blue function or graph that we see here, or configuration, is the correct configuration. The red is what's estimated from our model or our algorithm. And here we can see that this is dissimilar. And so this is one measure of how we can determine whether our program is working correctly, but there are other measures. And the measures would be, if we're looking at secondary structure, what % of angles were correct when we formed them? Is the chirality of the substructures correct? What are the number of constraint violations in our program? And what's the relative error of our torsional angles? Those are other measurements that are used to determine whether this particular program or algorithm does work. Now, where do I fall into this whole picture? It's a large picture, and we don't have a lot of time here, but my research is Nome. It deals with all of the same basic concepts that we've seen in the previous slides, but here again, the same fundamental problem with the choice of a nonlinear least squares approach, simple interatomic objective function, where the constraints are not part of the objective functions. Those are actually created outside of the objective function. And so it's also an atom-based approach. So what that means is when there's an update in the direction and the step length, that update is done based on an atom basis. Instead of updating simultaneously, the atom is done based on one atom at a time. Lengthy process, but what it does do is it allows us to be able to first identify, of course, the best local optimization method, because the local optimization method is really based on being able to compute the first and second derivative information. And if we're looking at a function that has 3 n variables, computing that first derivative, computing the second derivative, which we call the gradient and the Hasten matrix, is going to require a lot of time and effort, computational power. And so here, by using the atom-based approach, it allows us to reduce the dimensionality from a 3n dimension down to 3 dimensions and compute these quantities. And once we've done that, we're able to use a non-random perturbation method to be able to aid in global convergence. Ideally, what happens is once we have our local optimization, we have the step direction. We try to use a line search, backtracking line search, to be able to find the proper step length in the direction that we have chosen from the local optimization algorithm. And also using the line search, we can be able to satisfy some of the physical constraints that were not included in the objective function by using the line search. And so within this algorithm, there is a lot to show. This is just the basic input atomic distances, but all the other constraints are not part of the objective function. But there is a way to go through and look at to satisfy those constraints. Now, this may be a little bit much to go over here in this particular type of setting, but this is how the algorithm works. So we take a set, we set up a list of atoms in decreasing order of error, then we move the atom one at a time. So we start with the first atom, find its optimum position, and then we go and we say, okay, let's add on the second atom and find the optimum position. Once we have the optimum position for the first and the second atom, we add on the third atom and say, well, what would be the optimum position? And we continue this process until we find the best position for all the atoms in the protein, or until there is a stopping criteria is met. And so the move atom procedure is where we incorporate the update, determining the step direction as well as the length. And here we use a quasi-Newton type of approach, which is instead of actually computing the second derivatives, we use approximations of the second derivative to take care of some of the time that's required in updating our atom positions. And now for the local perturbation type technique, once we have found the ideal step direction, this is how we execute finding the global minimum based on the line search. Will it fall in this region or this region? And if it's part of this line search, we try to make it so that it falls in a region that still has low error, but that is, again, another way of finding the global minimum. The same happens with the different types of constraints. If we have an atom that's positioned in a place where it violates a certain constraint, the atom is then moved out of those positions here to a place where it still has low function error, but it does not violate any of the minimum separation constraints or chirality constraints or surface constraints. And here, the way that we determine the radii of the sphere is based on, again, the local error as well as empirical relationships from known protein structures in the PDB data bank. And so the validation and test of the structure. So the way that we set up a test and validation for this type of program is we construct synthetic data from known proteins, pairwise atomic distances, chirality information, torsional angles, surface data, and then we investigate what happens with certain proteins. So these are the 3 types of proteins that were chosen. And here is some quantitative results of what we look for. And that is, if we're given short-range distance information, if we're given 100% short-range distance information, we may not need constraints. It may just be enough just to use this distance information. But if our distance or structural information is insufficient or it is inaccurate, then we have to have those other constraints. And what these particular results show is that here, if I have 20% distance information and I add on an angle surface constraint, it still gives me a structure that's 2 angstroms. The way that we measure, again, the units for measurement is the angstroms. And so here it is 2 angstroms. If I have 40% of the data, it shows that whichever method I use is still going to give me a good estimate of the protein structure. And so these are all the 3 that we considered. And here we have the qualitative effects of including these different types of constraints and what happens when we compare the structures and see what's really happening here. When we look at the 2, they are similar and identical, but we notice that the substructure is not positioned correctly. And so that gives us a lot of information. But just by looking at the error of these structures, we see that the optimization has been satisfied, but the RMSD is a little larger in this area, and here it is lower. And so, again, this is just one way of, again, being able to determine whether the algorithm works the way we want it or should we add more constraints. And so modeling here at ODU, just to finish off with the last few minutes here, is where can we find out about modeling here? There are 3 different groups that actually look at protein modeling currently. And there's the green group that's led by Dr. Leslie Green. And there is also Dr. Passma's lab, which is led by Jennifer Passma from the chemistry department. And then there's Dr. Jing He, who also does protein modeling from the computer science department. And so they're currently actively working on protein modeling. For me, it's been a little while working on modeling proteins, but the ones that are listed here normally collaborate when it comes to work separately, as well as they collaborate on modeling proteins. And so with that being stated, it just kind of confirms a STEM type research project where it's not just one group of scientists working on a problem, it is many different scientists with different backgrounds working on a problem. And here, with that being stated, there are some references as well as websites. So thank you to REYES for allowing me to present. Are there any questions?
Thank you so much, Terry, for such a nice presentation. So there's a little bit of time delay. So people, if they have questions, it'll take a couple minutes for them to trickle in. That being said, we already have one, and I also have my own. So the first one that we have is from Anish Arade from Harrisonburg, Virginia. Sorry. What can protein modeling tell us about the organism/virus the protein are from?
Well, if you know the protein structure, then there is a way to determine from the protein structure the function of the protein. And once the function of the protein is understood, that leads into drug design. So that's kind of outside of the scope of my research, but I would think that based on what the structure is, there's a way that the protein can bind to other types of proteins that can cure different types of diseases.
Right. So knowing the structure of the protein is an input to be able to understand the role that it might play in other organisms. Right.
Absolutely.
Gotcha. Well, I had some questions. So I mean, as a applied mathematician, why? First off, what got you interested in applied math, and then what got you interested in this kind of research? I kind of imagine applied mathematicians to be, you have so many tools at your disposal and so many problems out there that perhaps are not directly what you're thinking about, that I would imagine that it's slightly overwhelming to try to figure out where to start getting involved. And so perhaps if you tell us a little bit about your path, I think that would be nice to hear about.
Oh, that's great. Ideally, I started off just wanting to be a math instructor, lecturer, just teach the concepts of mathematics. But when I got into grad school, I met up with one of my advisors who was into modeling biological aspects of mathematics, and one of those was protein modeling. So it was a choice between modeling cancer and modeling proteins. So because I had read one of my advisor's papers, I decided to choose the protein modeling aspect. And that particular person, Dr. Glenn Williams, knew a lot about proteins. His background was he was an engineer, but he has spent a lot of his time as a postdoc researcher scientist dealing with solving the protein folding problem. And I know I wanted to provide some information to some field of science. Am interested in medicine, but I knew that wasn't the field for me. So I wanted to find a problem that would relate to medicine in the math from the mathematical aspects and solve that problem or work on solving that problem.
Great. Interesting. So I see a couple more questions coming in. One from Joe Leo Chao from Chiayi, Taiwan. Sorry if I mispronounced your name and your city. The question is it possible for high school students to get involved with this? What are some prerequisites, what are some prerequisites that high school students need to know to be able to get involved in this kind of research?
I think the best thing for high school students is just to really take the science courses, all the courses to be taken seriously, but definitely chemistry, mathematics, as well as any type of coding classes that might be introduced in high school level, because those are important to the college level understanding of different types of research problems. And so I'm not sure at the high school level whether they have programs where you're able to get into solving protein type problems. But I know definitely at the college level, if you find a good advisor, and I think those are called REU type undergraduate research experience, and you can learn a lot about these type of problems based on the faculty in your department.
Great. Yeah. Actually, maybe if I could advertise a little bit, but some of the questions that you're tackling are things that in our Python for Physics, which is part of REYES, we've introduced similar concepts. So things about we've introduced the least squares functions, we've introduced minimization routines, and also we played around with the Ising model, which kind of has some similarities and flavors to the minimization functions, the energy functions that you were trying to minimize. So I think that's a good resource. I think if you're in high school and you want to get involved in this kind of research and you learn a little bit about programming, which is essential for this. So yeah, I mean, great response. I think we have one more question. Let me read it. So from Luis Angel Vergara Ortiz from Hidalgo, Mexico, who's been a very active REYES member. So thank you so much, Luis. Saying, thank you for the presentation. Have these techniques been used with the coronavirus analysis or something like that? If so, how?
That's a great question. My research for this problem stopped probably a few years ago, so I haven't quite kept up with what's going on with research in terms of protein structure modeling, but I'm pretty sure with what's going on with the pandemic that there's definitely a lot of research available. And if that student just types in protein folding problem or three-dimensional estimation of proteins, then it will come up with a lot of different types of projects that are currently being worked on as it relates to COVID.
Great. And maybe I'll save the last question for, well, okay, I have two more questions. One
Okay.
is practical. So these techniques, how well are they in predicting new structures of other proteins? So like, if you know the structure of one, can you reliably predict other ones by assuming that the coefficients that appear in the function of the energy is if you assume they're universal, can you then predict different structures from there, or is the prediction level not so great?
No, it depends. Now, there's been a lot of work done with protein structure estimation. So a lot of the different program softwares out there are very accurate and can predict proteins, but the problem is how many atoms are in the protein. If the protein has less than 300 atoms, then maybe that might be easier to predict the protein structure. But if it is a protein that has over 2,000 atoms, that might not be as easy to predict the protein structure. But there are different groups that are working on all different levels of estimating the protein structure, whether the number of atoms is a small number or a large number. So the one, I think, software that I'm aware of right now is called Rosetta, computational software for protein modeling. If the student types in Rosetta, and I can give the website as well after we finish, then they'll be able to go in and look at a software that actually is successful in predicting protein structures. That's just one of many.
Great. So we have one more. This is just a comment, just expressing thanks. This is from Shariclea Niku from Glasgow, Scotland. So hi, Glasgow. And maybe the last question, you've already touched on a little bit on this, but do you have any tips or recommendations for young scientists? So we have a lot of people from around the world that might be interested in pursuing these careers. Do you have any other tips that you would like to pass on to them?
I think the best tip for me as a student and now as a product of ODU is to find a good advisor. You want an advisor that truly understands the problem that they're working on, and they can actually explain the problem to the student. For me, I did not know a lot about biology or chemistry prior to actually working on this research project. I knew absolutely nothing. Never had taken a biology class. Chemistry, yes. Physics, yes. But that's one tip, that you always want to choose an advisor that understands the problem and that's active in research. Number two, the student want to do their part. If the problem requires that they know background, they have to know the background information, take care of the chemistry or the coding or whatever is required in order to be able to research a problem. And then the last tip, make sure you're willing to spend time. When we do research, research isn't just something we get in a day or two or a month. Sometimes some of the results take about 2 or 3 years. And so one would really have to choose, if they really want to be a researcher, scientist, or a teacher, really think about it as they work towards their degree, whether it's bachelor's, master's, or PhD.
Yeah, those are great tips. I think a good advisor can make a world of difference, and also you have to be really patient when it comes to research, because it can be a frustrating procedure, even though extremely rewarding when it works. So
yes, that is true.
Yeah. Well, thank you so much, Terry, for taking time out of your schedule to talk to us. Let me just remind people that since this is our last week, we still have a couple more Python for Physics lectures. We're going to start again tomorrow. Tomorrow, we're going to have a special lecture from Dr. Fatma Aslam, who's going to be giving us an introduction into quantum mechanics. And then we're going to have other activities planned. And then Thursday, we have our last REYES trivia night, where you're going to be tested. So we're going to select a few people to participate in a live session, but you're going to be able to interact with us via stream we're going to test your knowledge of the information that you've gathered throughout the last 8 weeks. So please join us. That's going to be a great deal of fun. And again, Andrew Dodson, our ODU physics alumnus, who's a famous YouTube star, is going to be hosting that trivia night. So please join us for that. Once again, thank you so much, Terry. It's been a pleasure.